Professor Helen Picton, and Helen's professor at the University of Leeds within the Reproductive Medicine Unit. And Helen trained at the University of Edinburgh. She did her undergraduate studies there and her PhD, and she's trained in all aspects of physiology and domestic species, and has been working on human oocyte development. And so we've learned a lot. She's been contributing to metabolism of the human oocyte and also in development of in vitro models. And sometimes when Helen and I are together, we're referred to as the hinge and bracket of the in vitro development world. So I'll leave it to you now.
Thank you very much for that introduction, I think. If you know who hinge and bracket are, you'll understand the problem. Thank you very much to the organising committee for the invitation to join this meeting. It's a really exciting programme, so I'm delighted to be able to contribute in some small way. We are required to declare any conflict of interest, and I don't have any for this presentation. The work that I'm going to be talking to you about has largely been funded through research councils in the UK, the Medical Research Council and the Biological Research Council. But I should declare, as Evelyn's already told you, that I am an ovarian physiologist and an egg and embryo biologist by background, and that will, I'm sure, influence what I tell you. So one of the things that we've been working on at Leeds in our research labs is markers of egg quality. You may ask why. Why do we need to understand markers of egg quality? Well, the situation is quite clear. Only a very, very small proportion of the eggs that we produce in assisted conception cycles, around 5% in fact, have the capacity for developmental competence. That means that they can mature, be fertilized, and go on and produce babies. And that is after you account for embryo freezing cycles and all of the other things that we can use to defer transfer. So there is a need to understand the biology of oocyte development and maturation, but also the inheritable legacy that the oocyte carries on into the next generation. And that's really the reason why we've been working in this field. I'm going to illustrate some of the tools that we and other labs are using to explore this concept of oocyte quality. And specifically, molecular and cytogenetic markers. I'll touch briefly on those. Time doesn't permit any further analysis, but it's important that they have a place in these discussions. And then move on to look at metabolic indices of some of these markers that we're looking at. And I'll refer to a combination of research in large animals particularly, but also particularly important in the context of this meeting, in human cells. So David Albertini illustrated this morning the importance of the association between the oocyte and its follicular environment. We know that the follicle, as it grows and develops, influences the oocyte, and conversely, we know that the oocyte influences the growth and development of the follicle. So it's a symphony, if you like. The follicle provides the orchestra, and the oocyte is the conductor of that orchestra. And that's particularly important as we start to look into metabolism, because what we're seeing is the coordination of metabolic processes between the somatic compartment in the cumulus cells and the egg itself, and the dialogue that exists between those two compartments. So here we have some pictures of a human oocyte going through meiotic maturation, and our oocyte with its investment of cumulus cells, either an oocyte from an IVF cycle or an oocyte with a compact cumulus complement that's going through an IVM cycle. And we can take snapshots of molecular indications of oocyte quality by looking at these different compartments. We can, of course, harvest cumulus cells from IVF cycles or from IVM cycles, and we can look at the molecular constitution of those cells. We can do targeted studies, and this is some of our own work from Leeds, looking for genes of note that we believe are important from oocyte development. There's some characterized here. And look at their expression patterns in oocytes that have the capacity to progress or the capacity to arrest in vitro. And you can see discrete differences in genes associated with oocyte development. Conversely, you can see genes which we know to be important in the signaling process to induce maturation in the oocyte, namely meiotic maturation and expansion and emulsification in the cumulus cells. And we can see differences in oocytes which have the capacity to progress and those that have no capacity to develop. But those are only targeted studies, and although they're very useful, they are limited. So what we've seen in the literature by our lab and many others across the world, some of whom are represented here in the audience today, that the recent literature has employed much more global screening approaches to look at the full complement of genes in eggs and cumulus cells. Microarrays have been used extensively, and now we're seeing the same sort of experiments carried out using RNA sequencing approaches. And particularly we can see these effects and analyses being conducted on individual eggs, but also embryos and cumulus cells. We can and are also now seeing pyrosequencing evaluation of the methylation status of key components of embryos and also somatic compartment in the follicles. And as you know, many of you are looking and using cytogenetic analysis of embryos for embryo selection or for pre-implantation genetic diagnosis, but you can also use that as a tool for looking at egg quality. So these approaches are very useful, but they generate an enormous amount of data, and it's actually quite difficult to decipher the functional relevance of those sets of gene analyses in terms of oocyte developmental competence. And you can see the sort of problems that we encounter illustrated here in this very nice model that was published recently by Marc-André Saurad's lab. Here we have, this is now a data set from multiple microarray experiments in cow oocytes, and you can see the impact of follicle size, chromatin status on the types of transcriptome data that is generated, and also you can see the impact of different stimulation regimes, again, on the types of transcriptome data generated from the same oocytes using the same technologies. So there is enormous biological variation between individual oocytes in a cohort from an individual patient, but also significant variation between patients because of their infertile etiology or the stimulation protocol they likely have been exposed to. So what we find with these global screening approaches is, although they are very valuable and generate a lot of data, there's a lack of consistency between studies. And it's actually very difficult to pick out genes that are consistently replicated as indices of egg quality from those types of molecular data sets. So you can use them, of course, to go back and look at key components of the cytoplasm, as we've heard so beautifully from Justin's talk. Genes associated with mitochondrial activity, mitochondrial inheritance, stressors associated with endoplasmic reticulum, and of course genes associated with egg and cumulus cell function and metabolism. So you can go and reverse engineer those array and sequencing sets and go back and do functional studies that are targeted. But what does that tell us? It still doesn't actually tell us what makes a good egg. Molecular studies are inherently destructive of the cells of interest, so they're not very useful in a clinical context apart from driving the science forward. We can use polar body biopsy to decipher whether the egg is chromosomally normal, because we are assuming that the chromosomal complement that we take out in the polar body is a mirror image of what's left in the egg. But does that mean that we know which of these two eggs has the most developmental competence? We know which is normal, chromosomally speaking, but we don't know which one is best in terms of its developmental competence. So the work that we've been doing in my labs in Leeds in collaboration with numbers of other people is to use a slightly different strategy. What we're doing is we're trying to look at a functional aspect, namely metabolism, and use the metabolism as a functional index. It's a non-invasive assay of cellular health, and metabolism we're using as the tool to try and catch this particular pigeon. So here we have a summary of the sort of things the reasons why we believe metabolism to be so important. In this cartoon here, you can see a cumulus cell and you can see the egg. And we've heard from Justin in the previous presentation how we can no longer separate the different constituents of the molecular and cellular composition of the cells because they are all interrelated. And that is illustrated very nicely here. In terms of metabolism, the egg is unique in that its preference for energy production is through pyruvate metabolism. In contrast, the somatic cells use predominantly glucose for their metabolism. So we can discriminate between cumulus and we can discriminate between cumulus and egg in their metabolism. We can also look at fatty acid metabolism and amino acid metabolism, and all of these factors will have a bearing on key functional parameters, whether it is the resumption and completion of meiosis, the epigenetics of the cell, or the mitochondrial replication and mitochondrial function within our important cell, namely our egg cell. So metabolism is very useful. Furthermore, importantly, we can measure it, and we can measure it at single cell level. We can measure uptake of substrates and nutrients from a microdroplet of culture media. We can measure production by a single egg into the same culture media, and then potentially we can relate it to key parameters of follicular development, differentiation, etiology, and infertility treatment. So we have a measurable tool which is non-invasive, which we can use to assess egg quality. So does it work? Well, this graph here shows in human eggs measurement of pyruvate consumption in relation to lactate production, so metabolic uptake and production into our culture media. And what you can see is that oocyte pyruvate production consumption increases, so lactate production, so as pyruvate consumption increases, so the waste product, lactate production, also increases. And that's in individual human eggs. So to take the idea that metabolism and body metabolism, and etiologies associated with metabolism alterations, such as PCOS, for example, we can look at those types of etiology, and we can look at the impact that they may have on the metabolism of the eggs from those patients compared with control women. So this is a typical graph showing energy metabolism. Here we have our candidate molecules, pyruvate, which is our preferred substrate in eggs, glucose, our preferred substrate in somatic cells, and lactate, the end product of those metabolic processes. And what you'll see is uptake is always illustrated by, from the media's consumption is below the zero line, and production into the media is above the line. And what you'll look at here is you'll see that we've got patients who are normal, control women in yellow, and patients from women with PCOS, the full syndrome according to the Rotterdam criteria, in blue. And you can see straight away that there are differences, not in pyruvate consumption by the eggs, but in glucose consumption. Now, I've already told you that eggs, human eggs, eggs from any other mammalian species, would not be expected to metabolize glucose. So there is a metabolic abnormality in the eggs from patients with PCOS. We see differences as well in the production of intracellular NADPH between our control women and, again, our women with PCOS. So there are metabolic lesions, if you would, in the eggs of women with a metabolic lesion in the body and those who have infertile etiologies such as PCOS. So cut a very long story short, by looking at PCOS eggs compared with controls, we see our pyruvate and glucose consumption differences, but we saw no difference in the potential for meiotic maturation between eggs from those different cohorts of women. And we saw that the chromosomes in the G group were most likely to be involved in aneuploidy and predivision, and that's what we would expect because those are the small chromosomes, and they tend to be an age-related increase in aneuploidies in that chromosome set. So what else could we see by looking at this type of data? Well, if we analyse the data in relation to egg chromosomal complement, so full karyotyping of those eggs, what we can see is that the eggs, which are normal genetically speaking, had a different pyruvate consumption profile to the eggs that were chromosomally abnormal. And you can see the reverse in terms of NADPH content. So chromosomal health, chromosomal genes, if you will have an impact, losses or gains of chromosomes will have an impact on the egg's metabolic capacity. So that led us to ask the question, can we use metabolic fingerprinting to discriminate between our two oocytes? Can we see whether they are different in terms of their metabolic fingerprint, and is that fingerprint associated with their competence to, in fact, mature, fertilize, and produce healthy embryos? So we asked that question, and that question was led by my long-term collaboration with Professor Henry Leese, as you can see here, who works in York, which is only a few miles away from where we're based in Leeds. Now, Henry, if you know him, a few years ago proposed what we call the quiet embryo hypothesis. And his hypothesis is centered around the philosophy that the embryos that have the most efficient metabolism are the embryos that have the greatest capacity to develop into blastocysts and to produce healthy pregnancies. Now, given that the cytoplasmic inheritance of the zygote is inherited from the oocyte, we factored into this series of ideas is that the actual cytoplasmic inheritance of the zygote comes from the egg, and therefore it's egg metabolism which is contributing to this metabolic philosophy. So we conducted a series of experiments. This is now using bovine eggs as a model in the first instance, where we cultured individual eggs in microdrops of media. We then harvested the spent culture media and analysed their amino acid turnover, because that's the central tenet of the philosophy of quiet embryo metabolism. Then we inseminated using IVF the oocytes, and we tracked individual oocytes through culture to the blastocyst stage. And we related then whether the metabolic fingerprint of the egg was linked to its capacity to be not only mature, but to fertilise and produce embryos. And this is what you can see from the bovine data. This is a series of amino acids here that we can analyse. We can analyse 18 amino acids in 4 microliters of culture media. And you can see the profiles for oocytes that had no capacity to produce blastocysts versus oocytes that were fertile and went on to produce blastocysts. And you can see that in this species there are at least 3 amino acids which are significantly associated with the capacity of the egg to produce a blastocyst in vitro. We then went on to look at human eggs, and this was published a few years ago. This is work by Karen Hemmings. This is now looking at the capacity of human eggs to progress, to mature in vitro. And again, we have eggs that were arrested at germinal vesicle, eggs that progressed to metaphase 1, and eggs that matured to metaphase 2 in the different colours. And we can see that there were numbers of amino acids that are associated with the capacity of the egg to progress to metaphase 2 in the laboratory. If we look at the overall metabolism, that's net depletion, appearance and turnover of amino acids, and we analyse the same data set in accordance with patient age, whether they were older, over 35, or younger, less than 35, you can see there are significant differences in metabolism of amino acids in these individual eggs between women of different age. So, to cut a very long story short, what we can say is that carbohydrate and amino acid metabolism by human eggs are significantly linked to numbers of parameters. Significantly linked to oocyte developmental competence, either their competence to mature or their competence to be fertile and produce embryos. We can see associations with oocyte chromosomal or genetic health. There are links with patient age, there are links with patient etiology, and there are also clear effects associated with the type of gonadotrophin stimulation regime that we use in our patients before the eggs were collected. So far, we've focused on eggs at the mature end of the spectrum. But what we know from the biology is that the biology of egg development and the acquisition of egg competence starts 4 to 6 months earlier with the primordial oocyte. And in the progression down this pathway, we see a progressive accumulation of the organelles, the proteins, the RNA that the egg needs to confer competence to fertilize and produce an embryo. So what about metabolism? Do we see a change in the metabolic capacity as eggs go through this developmental continuum? Well, the answer is yes. Now, this is some data from a couple of years ago, but I don't think it's ever been replicated elsewhere because it's very, very difficult to generate this sort of data. This is now looking at carbohydrate, energy metabolism in mouse eggs and follicles as you go through progression from these very early stages up to and through ovulation. And you can see here glucose consumption. This is follicle data, so there now is glucose consumption because the follicle cells are using glucose as the follicles grow in vitro, and we can see lactate production. If we look at the oocytes from those follicles, we can see progressive increases in pyruvate consumption as the eggs grow up to a maximum. If we standardize that per unit volume, we see that once they reach their maximum size, there's no change in their pyruvate metabolism. And if we look at oxygen consumption, again, we can see increases in oxygen consumption as eggs grow, and that actually is variable according to egg size. So what we can do now, using a series of quite sophisticated technologies, is to start to link together multiple components of these measures. We can measure oocyte metabolism. We can look non-invasively or invasively at intracellular NADPH. We can look at mitochondrial activity. We can look at carrier typing and chromosomal health. And we can conduct sophisticated molecular analyses. But we are doing those sophisticated analyses now, having led into them by studying metabolic indices as a first stop in the process. So where does that take us? Well, I'm going to use mitochondria as a very brief example of some of the things that we're now beginning to see, because it links nicely with what Justin's just told us. So this is the sequence of follicle development. This is now data from sheep, and we can map the changes in mitochondrial DNA copy number across the developmental sequence in the eggs that we're analysing. You can, of course, as we've seen from Justin's talk, measure mitochondrial activity, and this is an indication of mitochondrial polarity using JC1. And we can get an index of not just whether the mitochondria are there, how many are there, but how many are active. But by linking these measurements together, we're now actually starting to see some very interesting facts. So this is some new data from one of my PhD students, Chutima Topipat, who's been working with me in Leeds. And this shows data associated from sheep oocytes, which are from prepubertal oocytes or adult oocytes. So now seeing a clear effect of age on oocyte mitochondrial copy number. And if you look at this in more detail, you will see here, these are a time zero analysis, so immediately after collection. So these are germinal vesicle oocytes. Here we have oocytes after 24 hours of in vitro maturation, so these are M2 oocytes. And in the intermediary, we have oocytes cultured for a short period, this is only 6 hours, in discretely different types of culture media. And you can see a huge change in the mitochondrial copy number and also the variance in the data just as a result of the culture environment that the eggs are sitting in for that short period. And you can see that in the prepubertal oocytes, and you can see it in the adult oocytes as well. Now, what we're able to do now with our non-invasive assays is actually to measure metabolism and then link the metabolism to mitochondrial copy number measurements in the same cell. And this is what's illustrated here. Here we have our prepubertal oocytes versus our adult oocytes with mitochondrial copy number. And this is the glucose, pyruvate, and lactate metabolism from the same oocytes, again with the lamb data, the prepubertal data in blue and the adult data in red. And we can see discrete difference in pyruvate metabolism by the young versus old oocytes, just as we can see discrete differences in mitochondrial copy number in a controlled environment in vitro between the adult and the young oocytes. And if you start to process this data in more detail, you can actually see how the relationships change as these oocytes age naturally in the body. So this is the data now plotted out in terms of pyruvate consumption in relation to mitochondrial copy number for our young population of oocytes versus our old population of oocytes. And you can see the relationship changes as the oocytes age. And although I can't show you the data, because it didn't copy very well when I brought it into this slide, if you actually standardize the data through another set of calculations, in other words, you standardize the data for energy consumption according to mitochondrial copy number, so you divide effectively the pyruvate consumption data by the number of mitochondria, what we see is an exponential decline in metabolism. So the higher the levels of mitochondria, the lower the pyruvate consumption. So there seems to be an inverse association, regardless of age, between the number of mitochondria and the pyruvate consumption in our cells. And that's within the same individual cell. So to draw everything to a conclusion, it is now possible to conduct multiple assays of oocyte quality on the same cell. So we can start to make functional links between key parameters such as mitochondria, mitochondrial copy number, mitochondrial activity, energy metabolism, and parameters such as cytogenetics and indices of egg quality. And we can extend those measurements backwards along the developmental pathway of egg development, and we're starting now to build really complex portraits of egg development throughout folliculogenesis across different species, including human and sheep, which is our model species we work on a lot in Leeds. We can quantify indices of oocyte quality using non-invasive assays. Now, there's still a lot of work to be done in this area, but it has potential applications in the clinical context. What we see is that oocytes which have a different metabolic signature, which have no capacity to develop from those that have the capacity to either mature or to produce healthy blastocysts. We believe that oocyte quality in vivo and in vitro is informed by the oocyte cumulus complex and the presence or absence of the cumulus cells in culture, for example, will of course have a profound bearing on what we measure in our cells after culture. By manipulation of the sort of metabolic indices that we're seeing, we may well be able to reverse engineer our IVF protocols, for example, so that we can focus on the production of egg quality rather than egg quantity, and that might improve the outcome of our assisted conception treatment cycles. So with that, I'd like to just finish by acknowledging that an awful lot of people have been involved over far too many years in this type of project. It's very, very time-consuming. So we have current members of the lab, both in terms of academic input and students who are studying for postgraduate research degrees, and of course our friends and colleagues at the Leeds Centre for Reproductive Medicine, and my life collaborator Henry Leese, who's been enormously helpful in educating me in the ins and outs of metabolism studies, and Bruce Campbell, who's worked with me on some of these other projects that I've illustrated to you today. So thank you very much for your time.